Hi everyone. Today we'll be looking at a recent review paper on aging cell that discusses lifespan extending endogenous metabolites. This explainer will summarize the key findings from a 2026 review by Zhang and Han in the journal Aging Cell, focusing on the scientific evidence for specific molecules produced in our own bodies that may regulate the aging process.
Here's how we'll structure the discussion. First, we'll cover the general role of metabolics in aging. Then, we will examine specific classes, starting with amino acid derivatives and core energy cycle metabolites. From there, we'll discuss amino acid restriction pathways, followed by cofactors and signaling molecules. And finally, lipids and sugars, before we conclude with an overall perspective on the field.
So let's begin with the foundational concept. It is now a central tenet of geroscience that dysregulated metabolism is a core hallmark of aging. And at the center of this are endogenous metabolites. You can think of them as key signaling molecules that translate an organism's nutrient and metabolic status into specific cellular responses that ultimately influence both health span and lifespan. So what exactly are we referring to when we say endogenous metabolites? These are the small molecules produced by an organism's own metabolic processes. On a basic level, they serve as biosynthetic precursors and energy substrates, but they also play a much more complex role as dynamic modulators of signaling pathways and gene regulation, which is how they directly influence the processes of aging. All right. Let's now move into our first specific group of metabolites. We'll look at two amino acid derivatives, taurine and betaine, both of which have been investigated for their potential geroprotective properties. First, we have taurine. It is a sulfur-containing beta-amino acid that is synthesized from either cysteine or methionine. It's not incorporated into proteins, but is found in high concentrations in metabolically active tissues, like the heart and the brain, where it has roles in osmoregulation and calcium homeostasis. When we examine the evidence for taurine, a distinct pattern begins to emerge. In simple model organisms like the nematode C. elegans, supplementation has been shown to increase lifespan. In mice, benefits include reduced photoaging, amelioration of cognitive decline, and decreased markers of cellular senescence. But when we turn to human data, the picture becomes much less clear, showing mixed results and significant variability in its circulating levels with age. And this brings us to a significant controversy in the current literature. A prominent study by Singh and colleagues reported a consistent age-related decline in taurine across mice, monkeys, and humans, which led to the hypothesis that taurine deficiency is a driver of aging. However, subsequent analyses from other research groups which examined multiple large human cohorts found that taurine levels often increased or simply remained unchanged with age. This discrepancy calls into question the utility of circulating taurine as a universal biomarker of aging. Next, let's discuss betaine, which is also known as trimethylglycine. Synthesized from choline, it serves two primary functions. First, it's an osmolyte, protecting cells from osmotic stress. Second, it's a critical methyl group donor in one-carbon metabolism. In C. elegans, betaine extends lifespan by modulating conserved pathways like DAF16/FXO signaling. In aged mice, supplementation has been shown to improve skeletal muscle function and cognition and to enhance autophagy. But once again, as we look at the human data, observational evidence is inconsistent. Some clinical trials have even associated high doses of betaine with an increase in LDL cholesterol, and there are currently no randomized controlled trials demonstrating any anti-aging effects in humans. The proposed mechanisms for betaine in mammalian models appear to be quite multifaceted. It has been reported to upregulate transcription factors to preserve mitochondrial function, to stimulate autophagy via a specific methyltransferase, to blunt inflammatory cell death known as pyroptosis, and even to directly bind and inhibit the inflammatory kinase TBK1. So, what are the challenges in translating these findings to humans? There are several. First, as mentioned, the human epidemiological data are inconsistent. Second, gut microbiota can convert betaine to TMAO, a metabolite linked to atherosclerosis. And finally, high doses may elevate LDL cholesterol, which represents a significant safety consideration. Now, for our third section, we'll shift our focus to two central intermediates of the tricarboxylic acid or TCA cycle, α-ketoglutarate and oxaloacetate. Let's start with alpha-ketoglutarate, or alpha-Kg. This is a key intermediate in the TCA cycle.
In invertebrate models, supplementation with alpha-Kg has produced remarkably consistent results, demonstrating significant lifespan extension and a delay in age-related functional decline.
In fact, the evidence for alpha-Kg is quite strong across different species. Lifespan extension in both worms and flies is well documented. And importantly, this effect appears to translate to mammals, with a key study in mice demonstrating an extension of both lifespan and health span. The human data are still preliminary, but one analysis suggested a reduction in epigenetic age, and a clinical trial is currently underway.
So how does alpha-Kg exert these effects? The primary mechanism appears to be the direct inhibition of ATP synthase. This action lowers cellular ATP levels, which effectively mimics a state of dietary restriction. This, in turn, leads to the inhibition of mTOR and the activation of other pro-longevity pathways, such as AMPK and FOXO.
The other TCA cycle metabolite to discuss is oxaloacetate, or OAA. The proposed mechanism of action here involves altering the cellular NAD+ to NADH ratio. This shift can activate sirtuins and is another way to mimic a state of caloric restriction. However, in sharp contrast to alpha-Kg, the evidence for oxaloacetate in mammals is significantly weaker. While it does extend lifespan in worms and flies, a large-scale study conducted by the National Institute on Aging's Interventions Testing Program found no significant effect on the lifespan of mice. One potential reason for this translational failure is the molecule's chemical instability, which creates substantial pharmacokinetic challenges.
For our fourth section, we're going to shift our perspective. We'll now look at amino acids where restricting their intake, rather than supplementing it, is most often linked to longevity. Our focus will be on methionine and the branched-chain amino acids.
First is methionine, an essential amino acid. The dietary restriction of methionine is one of the most well-established and robust interventions for extending lifespan and health span, with positive effects demonstrated across a wide range of species, from yeast to rodents. What is particularly interesting here is that even though the pro-longevity effect of methionine restriction is highly conserved, the underlying molecular mechanisms seem to differ significantly between organisms. In yeasts, the effect is dependent on autophagy. In worms, it involves a metabolic interaction with the gut microbiome. In flies, it requires the transcription factor FOXO. And in rodents, it lowers IGF-1 and increases FGF21 signaling, but notably does not appear to involve mTOR inhibition. Now let's turn to the branched-chain amino acids, or BCAAs, leucine, isoleucine, and valine. They are well known as potent activators of the mTOR signaling pathway, which is a central regulator of cellular growth and aging. The role of BCAAs in aging is quite paradoxical. On one hand, lifelong restriction can extend lifespan in mice, and elevated BCAA levels are associated with insulin resistance and a pro-inflammatory senescent state. On the other hand, studies in yeast and C. elegans have shown that BCAA supplementation or accumulation can, in fact, extend lifespan. This is a significant conflict in the literature, which strongly indicates that their role is highly complex and context-dependent. In Section 5, we will examine a more diverse set of metabolites that function as cofactors or signaling molecules. We will begin with the gasotransmitter hydrogen sulfide.
Hydrogen sulfide, or H2S, is a gas produced endogenously by our own cells. In worms, the administration of H2S donors extends lifespan. In mammals, H2S levels are known to decline with age, and using donors can provide organ-specific protection. However, there is currently no direct evidence from controlled studies that H2S supplementation extends overall lifespan in mammals, which represents a significant translational gap.
Myo-inositol presents another complex picture. It appears to extend lifespan in worms, though different studies propose conflicting mechanisms. In the mammalian brain, it seems to play a dual role. It can support synapse formation, yet elevated levels are also associated with glial activation and cognitive impairment. As of now, direct evidence for a lifespan-extending effect in mammals is absent.
The biology of NAD+ and its precursors like NR and NMN is one of the most widely researched areas in the aging field. The preclinical data are certainly promising. In various model organisms, boosting NAD+ consistently delays age-associated physiological declines. However, the results from human clinical trials have been far more uncertain. While we can successfully raise blood NAD+ levels, the functional improvements have been modest and inconsistent, and some potential adverse effects remain a concern.
Spermidine exhibits remarkably consistent geroprotective effects across a wide range of species, from yeast to mice, primarily through the induction of autophagy. The human data, however, are complicated. Epidemiological studies have linked higher dietary intake with lower mortality, but other studies have associated higher plasma spermidine with markers of brain aging. A key challenge is that short-term supplementation often fails to measurably increase circulating levels of the compound.
In our final section, we will briefly cover several metabolites related to lipid and sugar metabolism before moving to the overall conclusions of this review.
Here we have a change of pace. Ceramides are generally considered to be negative regulators of longevity. These sphingolipid metabolites accumulate with age across species. Interventions that inhibit their synthesis or lower their levels have been shown to extend lifespan in worms and healthspan in mice. This aligns well with human data, where elevated plasma ceramides are a well-established risk factor for cardiometabolic diseases.
Next are the ketone bodies, particularly beta-hydroxybutyrate, which are produced by the body during fasting or carbohydrate restriction. Supplementation with beta-HB extends lifespan in C. elegans. In mice, ketogenic diets that were initiated at midlife were found to extend median life. For humans, the primary area of interest thus far has been in neurodegenerative diseases, where they may offer modest cognitive benefits.
Finally, there's a group of other molecules, such as vitamins and the sugar trehalose, where the evidence is either very mixed or weak. For example, trehalose extends lifespan in worms but can shorten it in flies. Vitamin D3 shows benefits in worms, but large human trials focused on frailty have been negative. The evidence for vitamin C and B12 is similarly inconsistent. The bottom line is that at present, there is no strong conclusive evidence for their role in extending healthy lifespan.
This brings us to the overarching conclusion of the review. While the preclinical evidence for many of these metabolites is compelling, especially in invertebrates, major translational gaps still prevent their direct application as human anti-aging therapies.
This schematic from the review helps to tie these diverse findings together. It illustrates that even though these metabolites are chemically quite different, they often converge on a limited set of conserved aging pathways, such as nutrient sensing, mitochondrial function, and autophagy. This convergence strongly suggests the existence of common underlying mechanisms in the metabolic regulation of aging.
This summary table recaps the general pattern we've seen repeatedly. For many of these molecules, there is strong evidence for lifespan extension in invertebrates. But as we move up to mammals, that evidence often becomes more limited, focusing on specific health span metrics, or it disappears entirely. And when it comes to definitive clinical evidence from human randomized controlled trials, it is scarce for almost all of them. And this leads us to perhaps the most critical takeaway from the entire review: context is paramount. The effect of any of these interventions is not universal. An organism's genetic background, for example, can determine whether it benefits from dietary restriction at all. Sex is another crucial variable. Restriction of the amino acid valine extended lifespan significantly in male mice but had no effect in females. And on top of that, fundamental parameters like optimal dosing and timing remain largely undefined for human applications. This all leads to a final provocative question for the field. Given the profound variability in response based on genetics, sex, diet, and other factors, is it likely that a single, one-size-fits-all metabolic supplement will emerge as a panacea for aging? The evidence suggests probably not. Instead, the future of this field is likely to be in personalized geroscience, where metabolic interventions are carefully tailored to an individual's specific biological context.